So first off, what a pleasure it has been to be here. Real juggernaut of excellent talks, really inspiring. So thanks so much for the invitation. Second thing to say is Lindsay's done most of my work for me. So I think we can just focus on some of the key concepts and I think emphasizing the role that genetics has to play when properly integrated into clinical translation for drug discovery and development. I think the way we see things is largely shaped by our experiences to date. So with this in mind, I'll give a bit of context to how I got to where I am now using genetics for drug discovery and development. People say, how long have you been in your current job? I would say I've been in this current job for around 20 years because all of the experiences to date have contributed to that. So by background, I'm a physician, trained here at Oxford for 6 years, moved to London as an academic scientist, with my research focusing on using genetic data to unravel disease mechanisms and inform therapeutic development. Published close to 300 research papers in that area, specifically how we can use genetic variants to mimic pharmacological perturbation of drug targets and inform clinical development. And that took me to Novo Nordisk at a particularly exciting time. I joined in the peak of COVID in 2020, and I saw it go from being a company most commonly mistaken for Novartis. So I told my parents I worked for Novo Nordisk, and they said, you're based in Basel now, to become the biggest company in Europe and changing the paradigm of how we treat obesity, how we consider obesity, and we've heard a lot about the power of GLP-1 this morning. I realized that Novo Nordisk was a fantastic organization, but the role that genetics had to play was far bigger than one organization. I realized that actually genetics is very likely going to completely change the paradigm of drug discovery and development, and I wanted to offer that more widely than one organization. I wanted to bring it to the whole the whole field more generally. So let's just take a step back and consider this in its most simple terms. Drug development is incredibly inefficient, dissatisfyingly inefficient, and I think the fact of the matter is if we take the US out of the equation, there is no innovation. The US is currently funding the whole world's innovation, and if we take that away, we're not going to come up with any new drugs. And the simple reason for that is it is simply too expensive. Right? It takes too long and it costs too much money, and that is because the approach we're taking probably isn't optimized. It hasn't meaningfully changed since its inception, and it doesn't look like it's going to do that unless we really go back to the basics in thinking about this paradigm. The majority of data to inform drug discovery and development come from animal studies. People aren't animals, and so we're always going to have issues about translatability. That's not to say animal studies don't have a role to play. They're pivotal for toxicology, safety, pharmacological evaluation. But in terms of deciding whether the drug works, it's simply not adequate to put it in the mice or even a non-human primate and infer efficacy for humans. The second issue relates to traditional epidemiological study designs. When you see associations between a risk factor or even a drug target and an outcome, that doesn't necessarily infer causation. There could be any number of other explanations, including reverse causation or confounding. And these two limitations alone have been one of the main reasons for the tremendous failure rate we've seen, more than 90%, because it's very difficult to infer causal effects of a drug target in a human. And that problem has persisted since the beginning of pharmaceutical industry, essentially. Now we're in a position where actually we can overcome these two major limitations, right? The first is causation, and we can overcome that because genetic variation occurs at the point of conception and so before disease processes. And secondly, these are human data. Large-scale human genetic data can infer causal effects of drug targets. Genes code for proteins, and the vast majority of drug targets are proteins, particularly for biologics and small molecules. And so we're at this position now where we have large-scale human data telling us about naturally occurring variation in the genes that code for protein drug targets. We have a natural randomized controlled trial that is telling us about what drug targets are doing. And people have actually become quite disheartened, paradoxically, about genetics because the promise of genetics has been there for 20 years. For 20 years we've been hearing genetics is going to change everything, genetics revolution is coming, drug development is solved, and it hasn't delivered at all. And part of the reason it hasn't delivered is because the data in its breadth and wealth hasn't been there until very recently. Over the last 5 to 10 years, we've seen an exponential increase in the availability of large-scale genetic data. That means for the first time, we really can get meaningful insight about drug targets. about variation in those drug targets and what that means for efficacy, biomarkers, secondary indications, adverse effects, heterogeneity, precision medicine. All of these notions and concepts people have been talking about for 20 years, we're finally there because the data have become available. And we're going to be hearing a lot more about the power of data and how we can most effectively use it. But for the first time in recent years, we are seeing the promise of genetics become real. So we're at this position now where we have a 10% success rate, put another way, a 90% failure rate, which is not sustainable. As soon as the money from the US stops essentially funding the whole world's R&D, we're going to have to come up with a better paradigm for doing this. And it's time now for actually us to embrace the full power of genetics in a meaningful and translationally relevant way to do this with most impact. So what is this paradigm? The real importance of randomization is that we can infer causality. We can infer that the intervention is what is affecting the outcome and it's not some other confounding process. And that is why randomized controlled trials stay pivotal to guiding clinical practice. In many ways, human genetics parallels this because we have genetic variation in the gene coding for a protein drug target and it is randomly allocated at conception. So in essence, nature has done a randomized controlled trial for us. We have all the data now being accumulated, curated, and shared for us to basically analyze it and infer what the causal effects of these drug targets are. And that can inform all stages of drug development. That can infer safety for phase 1, on-target safety. It can infer biomarker strategy, so we're finding the right dose, the right combination of treatments, and the right way of measuring them, as well as informing on mediating mechanisms. It means that we're targeting the right protein for the right disease for the right person. And it also means we're really capitalizing on repurposing opportunities, secondary indications. And all of that can be done imminently before we even get into a patient with large-scale data. It sounds very simple, but actually what the challenge here isn't the methodology. We've got very smart statisticians and genetic epidemiologists that are really making a lot of progress on that. We've got people that are accumulating masses of relevant data, multi-omic data, clinical phenotypes, endophenotypes, disease outcomes, heterogeneity, gene expression, cell-specific tissue, all of that is there. But what we really need now is to bridge all of this together. We need the data to link in with the correct methods, and we need that to be used meaningfully in a translationally relevant way to come up with effective outcomes for the right patient, and that needs to be bridged together seamlessly. That is where the challenge is now in the current era, to most effectively have impact with this and reduce the burden of suffering. All of this isn't to say genetics is going to replace everything else. It is 100% not going to do that. But the fact is, and that was one of the questions I would have asked Lindsay, about 30% of drug targets don't have genetic variation in the relevant protein coding gene. And that is because they are either too pivotal to the survival of the organism, embryological development or adult development, and so you don't see that variation. So the fact is that 30% of drug targets, you're not going to use genetics to discover them, and you're not going to be able to use genetics to inform their development. But for that other 70%, you can dramatically improve the efficiency, you can dramatically improve the discovery rate and the optimization of clinical trial efforts. So it really does make sense to capitalize on this when it can be done, and then integrate it. So you're still doing everything you would have done anyway, but you have this massive game changer of causal human evidence to help you prioritize discovery and development decisions. And I want to give a couple examples just to showcase this. So these are all works that I've been involved in, and I think they relate to real-life drugs. So the first is factor 11. Factor 11 is a circulating protein. It's a coagulation factor, and it's implicated in thrombosis, but also bleeding. It's been prioritized for development as a novel anticoagulant. So the blood clots, and that causes things like venous thromboembolism and cardioembolic stroke. There are existing effective therapies for this that target factor 2 and factor 10, but they are complicated in that they have an increased bleeding risk. So there is morbidity associated with their use. Factor 11 was prioritized as a promising target that has similar efficacy as the existing therapies, but a lower bleeding rate. And the fact is we can use genetics. A lot of the work for factor 11 was driven by genetics. We have a factor 11 gene. There is naturally occurring variation in that gene where roughly half the population have a variant that associates with higher factor 11 levels. And we can study that large-scale population data to infer the effects of having higher factor 11 levels or inversely having lower factor 11 levels. And what we find is that individuals that have higher factor 11 levels have an increased risk of cardioembolic stroke and venous thromboembolism. So we can use that insight to prioritize what are the subtypes of disease we should be using this to treat, what are the adverse effects, and where is it more or less likely to work. A really important point is that while the genetics can tell you whether a target may be safe and effective, it doesn't tell you about the particular chemical entity, and it doesn't tell you how it compares necessarily to the standard of care. So while the genetics and clinical data support efficacy of factor XI inhibition for thrombosis, clot formation, it doesn't necessarily mean that it's going to be better than the existing factor X inhibitors or factor II inhibitors, and it doesn't necessarily mean that we've got the chemical composition right. So genetics can dramatically improve the probability of success. It is not there to replace everything, and it doesn't mean we need to stop thinking, but when used the right way, it can dramatically improve our probabilities of success. The second issue, the second example is actually GLP-1R. So I published this while I was at Novo Nordisk. There is genetic support that GLP-1R is going to be efficacious for diabetes and obesity. If you go to the GLP-1R gene, you see a signal there for type 2 diabetes, you see a signal there for obesity. What's really interesting is it's a different signal. So that relates to the obesity effect being related to satiety in the brain, and the diabetes effect being related to insulin response and glycemic control in the pancreas. If you take those genetic variants that mimic the signaling effect of GLP-1R, you can use that to inform repurposing opportunities. And at Novo Nordisk, we were particularly interested in optimizing the design of the cardiovascular outcomes trial and whether heart failure should be included within that. At the time, there was some controversy. GLP-1 agonism increases heart rate. There was some clinical data that some of the existing GLP-1R agonists may actually worsen heart failure outcomes or worsen cardiac function, so it was controversial. The genetics was pretty clear. The genetic variants, the genetic instruments mimicking GLP-1R agonism were associated with lower risk of heart failure and improved left ventricular ejection fraction, and that was above and beyond what you would expect from improved glycemic control alone. So this study contributed to the data package that led heart failure to be prioritized, and actually some years later we see phase 3 clinical trial data strongly supporting efficacy of GLP-1R agonism in heart failure across subtypes. So again, an example of where you can even in very late stage identify novel indications to maximize the impact of a drug using genetics. And then the third example I want to give is interleukin-6 inhibition. So interleukin-6, for some time, its inhibition has been approved and is efficacious for inflammatory diseases like rheumatoid arthritis. But actually there may be other opportunities. Nature has offered us natural variation in the interleukin-6 receptor. There is a missense variant that changes the amino acid sequence, and we can use that variation using large-scale population data to infer the effects of pharmacologically inhibiting interleukin-6 inhibition. And there's been a few examples where we leverage this to prospectively prioritize clinical development decisions. The first was polymyalgia rheumatica. So this is an inflammatory disease that affects the vessels, can impair muscle function. We showed that genetically predicted inhibition of interleukin-6 signaling is associated with better outcomes. We published that, it was used to inform the trial design, and sometime after that we had clinical trial data that supported its efficacy, and now this drug is being used to help these patients. Another example of interleukin-6 was in the height of COVID. Rapidly scurrying, surging for effective medicines, steroids were shown to be effective, inflammation was implicated, so what anti-inflammatory target should we be pursuing? Again, we published genetic data that showed individuals that had genetically predicted inhibition of interleukin-6 signaling were at lower risk of developing severe COVID, of ending up hospitalized, of ending up in ICU. We published that, and some months later, the recovery trial supported those genetic predictions and showed that individuals receiving tocilizumab were less likely to suffer severe morbidity or mortality with COVID-19. Most recently, we've published a plethora of data implicating inhibition of interleukin-6 signaling in various aspects of atherosclerotic cardiovascular disease. So it's implicated in coronary artery disease, large artery stroke, abdominal aortic aneurysm, and we're expecting a readout of a phase 3 trial, the ZEUS trial, that is again being led by Novo Nordisk to explore interleukin-6 inhibition as a treatment for atherosclerotic cardiovascular disease and various other cardiovascular indications. So genetics is powerful, drug development is inefficient, and I strongly believe that appropriately using genetic data is going to transform the paradigm of drug development and inform every aspect of its development. There are now numerous examples that have prospectively demonstrated its power, but what is pivotal to its effective use is integrating the various expertise, the right data with the right methods, with the right question and the right translation. Without the appropriate integration of that expertise, it will never fulfill its potential, and that's what we really need to achieve here. Thank you.